Hello everyone and welcome to our NGS-CN Summer School online collection on spatial transcriptomics. Before we jump to our next speaker, let's take a look once more who we are. So it's a next generation sequencing competence network that consists of 4 production sites as you see on the map. So in 2019, DFG has funded centralized sequencing applications. There were quite a lot of calls, and within each call there were a lot of projects. And from 2024, DFG has changed the strategy towards individual sequencing applications. That means that now there is no special deadline for a centralized call, and you can co-apply at the DFG together with the chosen genomic facility. If you wish, here is a QR code, contact us, and then we could take a look and maybe sequence together. If you want to know something more about our news and upcoming events, then you also can take a look at the QR code, scan it, and get into our mailing list. Let's grow community together. And with that to be said, let's welcome our next speaker.
Hi. I wanted to start with this beautiful image and just ask, the question is, how do we go from our idea, our research idea, a sample, to sequencing reads, from the reads to gene counts, and from gene counts to an image like this, and where, every dot is a single cell that we sequenced. And how does an image like this help us to understand the sample, the question, and the heterogeneity of the cells in our sample? And this is the topic of my lecture today. So I would like to start with a little overview. We've heard parts of this morning. So we start with our question and a sample, and we sequence the RNA of individual cells with whatever technology. So we get millions of sequencing reads that then we have to map back to the genome. So the first part was the talk from Susanne, and the second part, the talk from Andreas. So here, what we are doing is basically take all these millions of sequencing reads and we map them back to annotated genes in the genome, and we count how many reads map to which gene. From there, we derive a huge table. For example, we have 10,000 cells, so 10,000 columns, and 20,000 genes, which means 20,000 rows. And just one item, one cell in this table could mean, this gene has that many reads mapped to it in the experiment. We do this for all cells, all genes, we get this huge table, and this is actually the basis for data analysis. What we can do, or what we have to do, is then pre-processing. So we're actually cleaning up the data first. So then afterwards, we can get the best analysis possible. For pre-processing, we'll do quality checks, normalization, we find variable genes, we do scaling and dimensionality reduction. For a basic analysis afterwards, we will cluster cells, visualize them in 2D space, like the image I started with, and we try to annotate the clusters. In an advanced analysis, you can then go on and, for example, look into cell-cell communication, trajectory analysis, or add additional experimental data. Today, I will focus on the first 2 steps. So the learning objectives for today, for my lecture, is for you to give you, again, an understanding of the value of single-cell sequencing, understand the data analysis workflow, so how we go from raw reads to gene counts, data pre-processing, and basic analysis to cluster, visualize, and annotate the cells. And then I will give you some resources for further learning. Okay, so this is a bit of a recap from Susanne's talk. She also talked about the smoothies. It's always a nice analogy because, you can imagine you drink a smoothie and you have a taste like strawberry, but you don't know what else is in there. Like it could be actually half of orange juice, but you don't know. With single-cell sequencing, we want to look at the individual ingredients. So a bit more scientific. We see on the left, we have a bunch of cells. And then with the classical bulk analysis, what we would do is to pool everything together. And then we would get a gene expression profile for all the cells in the starting material. And this will be dominated by the most abundant cell type, so in the bottom. With single cell analysis, we will look at every cell individually and derive gene expression profiles for every cell. And then we can use these to compare them to each other and actually relate the cells to each other and learn about the heterogeneity in the data. Single cell sequencing is now used in many fields of research. Most often in my collaborations, people want to use it to identify cell types in the sample. You can use it in oncology, for example, to understand the heterogeneity of tumors, as they usually consist of different cell types, and they can play different roles in the development and also the resistance to therapy. In developmental biology, people use single-cell sequencing to look at cell lineage relationships. So how cells develop, from one state to another and so on. So you might question, well, if single-cell sequencing is so great, why actually are we still using bulk sequencing? And so I just give this question to you, to the audience. I have some more questions in the lecture. So maybe someone of you would like to answer, why are we still using bulk sequencing? It's just simpler, and sometimes it's enough. Exactly. So let's move on. How do we go from raw reads to gene counts? That's the recap of Andreas' talk. So in our group, we mainly use these two technologies for single-cell sequencing: SmartSeq on the left side, which is plate-based, and 10x Genomics, which is the name of the company, a droplet-based sequencing on the right side. The difference, as you heard before, is with SmartSeq, cells are pre-selected into wells, by fax into wells. So one cell will be in one well, and we can process them individually in parallel. With 10x Genomics, everything is mixed together, cells and barcode beads, and they are encapsulated into droplets. And because of the cell barcodes, we can basically sequence everything together, and afterwards we will know from which cell a transcript came from. The libraries derived by either technology will then be sequenced on an Illumina sequencing machine. I just show you the picture of our current NovaSeq 6000 machine. With SmartSeq, we are sequencing the complete transcript and only, like, let's say 1 to 600 cells, so rather small amount of cells, and we sequence them quite deeply. For 10x Genomics, we sequence the 3' end of the transcript, so just a part. But we can sequence, let's say, 10,000 cells, and we sequence them more shallow, so not as deep. So we've seen in Mac's talk how Illumina sequencing works. What I like to do from time to time is just go to YouTube and watch some of these videos again, because they're actually quite nice to learn how this works. With the current NovaSeq 6000 as for flow cell, we can sequence 12 billion molecules in one run. So that's really massive. That's a lot of data we're getting out. Basically, the sequencing reads, no matter whether we get them from SmartSeq or 10x, whether we map the data ourselves against the genome, for example, using STAR, or use CellRanger, what we get in the end is a gene counts table. Here is just a little screenshot from a gene counts table. Let's say we have 20,000 genes, 10,000 cells, the genes in the rows and the cells in the columns. So if we, for example, pick CD3D in row number 3, in the first column, so the first cell, we see that we actually found 4 reads that mapped to this gene in this cell. Okay, so this is important to understand what the gene counts table means, because it's actually the basis for everything that follows. do you see anything which is, let's say, special in this table? Because actually it's different from bulk data. Just look, at the numbers and see, do you think something is weird? So we have a lot of zeros. So we are working with single cells, so low starting material. Then we don't sequence really, really, really deep to have everything. So actually we have a lot of zeros in our data set. And the software we are using or the tools we're using need to be able to handle that. Okay, so we are using actually often different tools as compared to bulk RNA sequencing data. Okay, so now we actually have a gene counts table and we can get started. And that means we start with preprocessing the data. If you ask me or you ask another bioinformatician, the exact steps will probably differ a bit because, everybody has his way to work with data. But I believe that these are the main steps that we all have to do. So first, we always look at the quality of the data to remove low quality cells. Then we normalize data to remove cell-specific biases. After that, we can determine highly variable genes, so genes that vary a lot across the cells, because we want to include the biological signal while excluding the noise. Then we have to scale the data to remove gene-specific biases. We do dimensionality reduction to reduce computational overhead and noise. And optionally, we can remove batch effects. And I will walk you through these steps now. So first, quality control. So on the top left plot, what we're seeing is a distribution of the number of genes we can detect in a cell. Imagine the gene counts table again. For one cell, so one column, how many genes have a count more than 0? So at least one read. Okay? And then this is a so-called violin plot. It's basically just a distribution. We have two samples. We have PBMCs sequenced with 10x genomics and PBMCs sequenced with SmartSeq2. You can tell from this plot, for example, that the majority of cells sequenced with 10x have an average of, let's say, 1,600 genes found per cell. Whereas with SmartSeq, here we have an average of around maybe 2,400. Okay? So that's expected because I told you for SmartSeq, we're sequencing a bit deeper, so we also find more genes per cell. Why am I saying this? We are looking at the number of genes we find per cell because that's an indicator for low or high quality cells. If you imagine cells are dying or they are damaged in the sequencing process, we actually only capture parts of the material. So then the consequence is that we have a low amount of counts for this cell, and then of course also a low amount, or very likely a low amount of genes detected. So a low amount of genes detected means it's likely a cell that we can flag as low quality. A second measure that we're using is the percentage of mitochondria reads. Mitochondria have their own membrane, and when the cell is dying, this is one of the last to degrade. So proportionally, we will pick up more mitochondria reads. So we are looking for cells with a higher percentage of mitochondria reads. Okay. Looking at the distributions here, we can see that we find a higher percentage of mitochondria reads in the 10x sample as compared to SmartSeq. And why that is, I don't know, but it's an observation for now. Since we have two measures, we can plot them against each other in the scatter plot on the top right. On the x-axis, we have the number of genes found per cell, and on the y-axis, the mitochondria percentage. Every dot corresponds to one cell. And what we're looking for are basically cells over here that have a high percentage of mitochondria reads and the low number of genes detected. Okay, is what we call, or what we flag as low quality cells. To be honest, at this point, I don't remove them yet. I always like to keep everything in for a first round of analysis to see what's in the data. And then later I come back and revisit the quality controls. Okay. In the second step, we need to normalize the data. That is because the number of sequencing reads per cell is actually influenced by technical differences. So when we compare gene expression profiles of cells with each other, we might find differences that are not because of biology, but because of technical sampling effects. So let's say we sequence one cell much deeper than another cell. Okay, then this is something that we have to take care of before we can compare the profiles. There are many different tools out there or methods out there to do this. And a very simple one is called count depth scaling, or also log normalization. Here, the idea is that each cell initially contained an equal number of mRNA molecules. And that's what we try to reproduce. So for one cell and one gene, we take the number of reads and divide it by the total number of reads in that cell. Let's say for one column in the gene counts table, we go through it, and every number we divide it by the total number of counts in that cell. Okay, so then in total, when we do this, and after the division, we multiply it by a factor of 10, for example, 10,000. That would mean after we do this for a cell, the total count would be 10,000. So have a look at these two plots. So the left plot would be before normalization. So we have some cells with a high number of counts and some cells with a low number of counts. And afterwards, they will all be at 10,000. And then we log transform the normalized data so we can more easily work with it. Our gene counts table has, let's say, 10,000 cells and 20,000 genes. And we know that actually 1,000 to 3,000 genes are enough, with the highest cell-to-cell variation, are often enough to describe the global structure of the data set. So we are actually interested in genes that vary between cell types, because they will also help us to identify cell types in a data set, as compared to genes that, you know, are like housekeeping genes that are equally expressed everywhere. They don't help us to see different cell types in the data. So the goal is to include the interesting biological signal and exclude noise. And the good number is often between 1 and 3,000. In the second step, we have normalized the data so we can compare gene expression profiles between cells. But we can still not compare gene expression profiles between genes. And one reason is that genes have different lengths, and dependent on the technology we use for sequencing, they get a higher number of reads by definition. So what we can do very easily is a seed transformation. So afterwards, every gene will have an average expression of 0 across all cells and variance of 1. So that's very useful, for example, if you want to visualize a lot of genes in one plot, because then they are all on the same scale. Okay, so we're down to, let's say, 3,000 genes, but we know that actually we can still go down more and explain the biology of the dataset by far fewer dimensions. Imagine that you visualize the data in a high-dimensional space where the axes are genes. And now we know that actually we can try to find new axes, new dimensions that still can explain the structure of the data set. And these new dimensions are called principal components. So I will not explain how principal component analysis works. If you want to learn more, again, I would say go to YouTube or search the internet. They are really good pages. But basically what we do is we find new dimensions principal components, and they are sorted by the amount of variance they explain. So the first principal component explains the most variance in the data set. The second principal component, the second most variance, and so on. So now we can rank them or visualize them in a plot like this. This is called an elbow plot. And we know that the genes that change, in a coordinated way between cell types, they will be contained in the top principal components. And the genes where maybe one gene, changes randomly in one or two cells will be contained in the later principal components. So the question is, how many principal components do we take along in our analysis? And what you have to do here is basically look for the point just visually where adding more components will not help you to explain more variance in the data. So here I would like to give this question again to you. What would you guess is a good threshold here for the number of principal components? I went for 30 here, but you could also, I guess, try 10 or 20. So, I mean, anything above 10, I guess, could work. What's recommended is that if you're unsure, you take a higher number, so you rather error on the higher side than on the lower side. So you include some noise, but you're not excluding biological signal. So that's why I went for 30. And if you look really closely to the plot, you would actually see it because there's some lines missing. So now we have our data set, 10,000 cells and 30 principal components, and that's what we are working with. So we can now go from clusters to cell types. First of all, we would like to visualize our data set in 2D by UMAP. The idea is that we look for similarities in the gene expression profiles in a high-dimensional space, and then we project this knowledge into a 2D space. That would be the result for the data set we're looking at. Remember the cells, PBMC, sequenced with 10x and SMART-seq. And we see some, let's say, clouds in the UMAP space. So again, every dot is a cell. So UMAP helps us to find different groups of cells and also transitions between these groups. Well, not in this case, but you can have that. And ambiguous regions. What we can now do, because we have a 2D representation, we can color the cells based on the qualities we know. For example, the sample they came from, so the 10x or the SMART-seq sample. And here you see that the cells colored in gray and in kind of pinkish nicely overlap. And actually, the secret here is batch correction. There are some things to take care of when you look at the UMAP. So the parameters influence the plot, so you may need more than one. And the UMAP cloud sizes and the distances between them may not be as meaningful as you think, because the method has a local notion of distance. So that means, for example, going back to this UMAP, the cloud on the top left is not necessarily, the cells are not necessarily more similar to the cells in the middle than to the cells in the bottom right. Okay, this is not what you can say from this UMAP. Now that we have a UMAP, we also want to group the cells into groups by clustering. So basically the idea is to find cell type specific clusters, and then we can use them to find marker genes and to actually identify what the cells are in the clusters. We often use graph-based clustering for single cell data, where we first build a k-nearest neighbor graph. The nodes are the cells in that graph. And then we have edges between the cells with similar expression patterns. Okay, so it's a connected graph. And then in there, we actually look for highly connected modules. Those are the clusters we're looking for. Again, we have obviously parameters that influence the clustering. And the most important one maybe is the resolution, which determines the granularity of the clustering. A higher resolution value leads to more clusters and vice versa. And you actually need to optimize this in for every experiment, for every analysis. So I think clustering comes with some challenges. You need to determine whether your clustering and your resolution is appropriate for your data set. You need to identify poor quality clusters. In a way, you need to remain patient because you have to do this all over again. And then in the end, you want to identify cell types by exploring marker genes. In our case, this is what the clustering looks like. So the colors refer to the clusters. This is just some default resolution values. So taken together, UMAP projects high-dimensional data into a low-dimensional space to reveal the underlying structure. It's a continuous visual projection. Clustering groups the cells based on their similarities. So these are discrete groupings. In general, I think they mostly agree with each other, but especially in biological complex areas, they can also disagree. And with the UMAP, we might find gradual changes that we cannot see with the clustering. So now I told you I'm coming back to the QC later on again, and now is the point. Now we have clusters, and I can check again, do I find any low quality clusters? So for every cluster, I can plot the QC measures we have determined in the beginning. So on the top, I plot the number of genes found per cell, and in the bottom, the percentage of mitochondrial reads. The UMAP, the cells are colored by the respective numbers, and then we have the violin plots for a bit of more of a numeric feeling. So I'm asking you, what do you think, which cluster would we have to remove from the analysis? Cluster number 7, a low number of genes found and a higher percentage of mitochondrial reads. So maybe you can also suggest what are the cutoffs that we should use. That's what I would do also, 20 for mitochondrial reads. And I guess here for the number of genes, I would have taken also 800 or something along those lines. Okay, so I actually did that and I repeated the analysis. And these are our new clusters and the new UMAP. So now actually, what are the clusters? What is the identity of the clusters, right? We want to know what the cells actually are. There are 3 things we can do now. We can determine cluster marker genes for every cluster. These are genes that are especially up or down regulated in one cluster compared to all the other clusters. And then we can look at the function of these genes and infer the function of the cell cluster. We can obviously take marker genes that we already know because we know the subject, the research subject, right? And color them on the UMAP. And also more importantly, nowadays, we can map known references onto the data and predict the identity of the cells. Good clusters typically lead also to good marker genes. So the question is, how can we assess whether we have good marker genes? I like to use a plot like this. It's a dot plot where on the x-axis, we have the different genes I found computationally. And on the y-axis, the 9 different clusters. The size of the dot means what is the percentage of cells in the cluster that express the gene. And the color means how highly it is expressed. And I should say it means it's a scaled plot. So if a dot is yellow, it means it's higher expressed than in the other clusters. It actually doesn't mean it's highly expressed. So that's a difference again. But what we're looking for are basically clusters like 5, 6, 7, 8, 9, where we find a bunch of genes that are specifically expressed in the cluster, and they're higher expressed in that cluster compared to the rest. On the left side, on the contrary, we find, for example, in cluster 1 and 2, the genes are kind of overlapping, right? So they're expressed in both of the clusters very similarly. And also cluster 3 and 4. So here, if I would do an extensive analysis of this data set, I would actually go back and try to improve this. First, the plot is generated in R, and the analysis behind is with Seurat, and they have some functions to find cluster marker genes. Here, I think I used a Wilcox test where I test which genes in cluster 5 are differently expressed than in all the other clusters. And then I pick the top 5 per cluster and I visualize them, okay? Now, what's the statistics to say this is a good cluster? It's just visual. I look at the plot and I find 1, 2, 3, 4, 5 genes. I mean, all the top 5 genes are basically almost only expressed in cluster 5, highly expressed in the cluster. Whereas here on the left side, so you see we have a yellow dot, but we have nothing in the rest of the clusters. That's what I look at. And here, cluster 1 and 2, the genes that come up in cluster 1, they also come up in cluster 2. And they're not higher expressed in cluster 1 than in cluster 2. So I think here, maybe we have 2 clusters that could be 1. So just to show you again the UMAP, cluster 1 and 2 are here at the top. So maybe that's actually 1 cluster, okay? And cluster 3 and 4, I mean, they look quite different here, but who knows, you know, it's something that I would look deeper into if this was a real analysis. So in summary, I think these look good, these look not so good, but I move on anyways. The question now is, let's assume these are good marker genes, we want to move on. How do we now know about the identity of the clusters? What we can do is take the marker genes for one cluster and then look for the functions of these genes. And obviously, you can do a lot of reading, but there's also a lot of systematic catalogs that you can use. And you can use tools to ask, is there any functional term that these genes are annotated with more than you would expect by chance? That's the question you're asking. And then I went to Enricher, which I like to use because it's very simple to use. You can use it in R or also online. So this is the website. Just pasted the 5 genes. And then I look at cell types. And you don't even have to read much about it, but there's a lot of B cells coming out of it. So my first suggestion is that cluster 5 is a B cell cluster. Then I went to Google and say, okay, give me, B cell marker genes, and got these 2 out. So I visualized them on the UMAP. And again, you see cluster 5 is higher expressed than in the other clusters. And the third option that I told you is to map a known reference onto the data. And I used here azimuth in R. And basically, again, I get a prediction that these cells in cluster 5 are B cells. So we have 3 ways of looking at the data. And in this case, all 3 ways suggest that we have B cells over here. And then if this is, a real analysis, you would do this obviously with all the clusters in your data set. So in this point of the analysis, I usually hand over back to my collaborators, and I always encourage them to explore the data themselves. And the tool we usually advise to use is CellXGene, because you can load our analysis into this tool and then actually explore, just select some cells here, some cells there, and compare them with each other, find differentially expressed genes. So if you ever have the chance, you can give this a try. What we do often is to look into the single cell best practices book, because, just for us to look up because it changes over time also what's best to do with a single cell analysis. Then there are toolkits out there that you can use. I am an R person, so I decided to use Surat. If you're a beginner, especially in R, then actually Surat offers a lot of tutorials where you can get started with. So I quite like that. In our group, we also wrote scRNA-seq 2, which is like a wrapper around Surat, where we added a lot of documentation and a lot of visualizations in addition. And that's on GitHub if you would want to contribute or use it. If you're not an R person, there are also alternatives. For example, ScanPy, which is based on Python, and it's also very good. Oscar, but I never tried this one. So this is, it's just from the website of Surat. You see there are different tutorials. And just start with the top left one if you're new to single cell analysis and try to reproduce what they have. A screenshot of our scRNA-seq 2 workflow. And so in summary, I hope I could repeat again for you to understand the value of single-cell RNA sequencing and help you understand the data analysis steps from raw reads to gene counts, the data preprocessing, and basic analysis, and some resources to learn more. As an outlook, the steps that I explained to you, especially the pre-processing steps, which were a bit lengthy, but also the clustering, the UMAP, these are steps that we can also universally apply to spatial data. I mean, we also have spatial information that we will use, but we can also use many of the single-cell analysis steps to spatial data. And I have actually done this here to a spatial data set. So we have a UMAP, just like we had it before. But then actually we can, color these clusters also in the spatial context. And that's what you will hear about tomorrow. And also in the hands-on part next week, we will look into this in detail and actually do this together. If you, want to reach out later, you can use our contact details.